Recording.
So you just record the screen. You record the screen.
Okay.
So we need the camera as well.
Take it on.
Maybe we should put it down.
That's from my group.
Yes. Encouragement from
From
them.
my group.
Yes.
I am your lecturer from now on, so you should have to keep silent. Actually, I was somehow lectured, so I can't. I think I can.
Clear your pressure.
Although, how many are these? 5
in progress.
Right. Yeah.
Actually, I need this. So
Recorded.
it's okay. The recording is going is here. Okay. So can I see the scene before he start?
Right.
I just want to check this here. Okay. So you should move from here over there, and please fill all the chair. And let me ask others to come and join.
Five minutes?
Wait.
My name is Mohammed, and I am the presenter of Good Faith. And I will share with you here our contribution from the case study that yesterday. So I will start, and then after I complete my presentation, I think questions will be there. So you can ask me, and also you can ask the participants of the group. Let me start. The case title was Emerging of a Febrile Illness of Unknown Caused Among the Population and Cities of Upper Egypt. That was like case title, and we are group 3. This is the main goal of the case study. The main goal of case study was, the goal of this case study is to understand the challenges faced by those involved in surveillance systems, highlight gaps, and explore improvements to disease surveillance system. This was the first question. Factors that make Aswan River an interesting area for epidemiological investigations. As you have seen, the map of this place, Aswan and Aswan, it is a place between Egypt and also Sudan, and it has many factors which are contribute to be interestful for investigation. So these are some of them. The first one is cross-border mobility linking Egypt and Sudan with active population movement. The second, presence of agriculture and tourism, including close contact with igneous forces and a hot, dry climate with vegetation, canals, and drainage ditches as an aerial mosquito bleeding site. High density of tourists and local workers expose water, bodies, and animals, which might come with newly emerging infections. Mixture of urban and rural settlements enabling zoonotic and vector-borne disease spread. The second question was design a case investigation data from follow-up on the onset of disease, expert related to information and the time of sample taken show details of affected cases and how they may relate to the sent samples. The first one, patient demographic data, including name, age, address, occupation, clinical data, onset of date, symptoms like fever, also severity and outcome. Exposure history, animal contacted travel, mosquito exposure, water sources, sample collection, the date that the sample has collected, the type of sample, as well as sender and storage conditions. The other one is, okay, the other one is December 2002, 5 people near Lake Nasir developed fever, headache, and weakness after a contact with horses and mosquito. Blood and CSF samples were sent to the Ministry of Health Virology Department. Timing and exposure links suggested a common zoonotic or factor-borne, possibly Rift Valley or Western, and that was our guess after we assessed the conditions. Laboratory findings case classification like suspected or confirmed. First, the case has been suspected and it will be confirmed with positive or it will be confirmed with, or maybe it will need some additional investigation so it will remain suspected. Contact list, this is the graph that the questionnaire that we can use to collect this data. The third one was, what are the clinical and post-mortem investigation that are applied to disease to diagnose the cause of disease. Clinical testing including full blood count or CBC, liver enzymes, CSF, MRI for detection of encephalitis. Postmortem was histopathological investigation is including liver, lungs, brain, and intestinal tract. For virological investigation, PCR, real-time PCR was included. In case of toxicological, or maybe if the person is toxic, if it's necessary, also it can be done. What are the recommended samples to collect it from the patient according to the mentioned symptoms? After we assess it, this is our conclusion. Maybe blood, CSF, urine, and stool. Maybe histopathological investigation is also needed. Describe and classify the different suspected cases that may lead to a febrile illness symptoms of these cases. So the infectious cause may be arboviral, like West Nile virus, Rift Valley fever, dengue, maybe, or maybe it can be bacterial infection, or maybe parasitic infection. What are the possible ways of transmission of this unknown disease? The possible ways could be vector-borne. It could be like bites of infected mosquito. Zoonotic cycle maybe from birds to mosquito to humans. No direct transmission was not investigated, like directly from human to human. What type of packaging should be used to transport the samples and why? We need to organize transportation of blood samples and tissue. So this is transportational, or how we can transport and what kind of packaging system that we will use when we are transporting this sample. We should have to use a triple packaging system according to the WHO guidelines 2015 to 2016. And as you aware of, or as you familiar with that, the triple packaging is subdivided into this. Also, we can also maintain cold chain. If we are using PCR and sending pure DNA, we should have to maintain also the temperature. So it could be included. Which type of surveillance technique do you think must be activated and conducted during the approach of these cases, and why? We have concluded active surveillance for febrile or neurological illness because of the person was also having some neurological symptoms. Event-based surveillance for animal and pet. That is like we can collect some samples from the birds and the animals which have been affected maybe, or we can get from near the place of this incident to further assess whether it is from zoonotic or not. Also, entomological surveillance to monitor mosquito density and species. The ninth one, do you consider this disease outbreak as a severe? How do you define this and why? Yes, we concluded yes. And the reason is because high case fatality rate among children and elderly. Neurological complication and deaths within days has happened in this case. Wide geographic spread because of S1 has links with these places, and there are a lot of tourism is coming to that place. So maybe the disease is from another place to here. Therefore, according to these findings, we should have to consider it as a severe outbreak and require national and international interactions. Review and complete this sheet. I think everybody has seen a sheet on the PDF, or maybe if you have the pages. So that was the conclusion of this is the conclusion of that graph or table, that table. It has some missing places, so it was supposed to fill it. So we have done it. This is the summary. The summary of report cases, we have summarized it as a West Nile virus breakout. The total human cases was 130, majority from Aswan Governorate. Affected groups was like this: 73 was male and 57 was females. The children who was having this disease was also like they were having this age 5 to 15, and adults was 18 to 45. Severity, it was mid to severe. That is mainly among the elderly. Some deaths have happened, and it was all about the patient disorder. The dead bodies were above 60. This is the laboratory findings. TSF and tissue was positive. So for W and V, West Nile virus. Okay, lineage cluster around. There was clusters of people from other places like tourists, so it could be linked to that. Like the disease, we can say the disease is from another place, or we may guess the disease is originated from here. That can draw an epidemic curve according to its shape. Try to assume the pattern of the infected spread the population, like pointing, intermittent, and propagated. What is the importance and characteristics of epidemic curve? Epidemic curves. Epidemic curve interpretation. Initial cases has started December. Peak around, or the most, the time that the disease frequency was high was December, and the deadline, the decline of the disease has decreased after mid-December. But propagated epidemic curve. Epidemic. So it was vector-borne spread, like mosquito was the factor who was spread. The vector-borne. Importance of epidemic curve is these are the importance of epidemic curve because of they are asking why this is important. So the importance shows the time and case I have here. So you can see it. We have derived this from this information which I was filled. So we concluded this graph. This is case detail, and it is the epidemic graph. I think you have remembered that when we were taking this, the lecture was explained, and some of the participants also has asked what is it, and I think he showed us a picture like this. So maybe it will be similar to this, or maybe it will be different because of, I think that was too much dense, but this is spread through that. So you can read again if you need. Identify who who are close contacts and what is their role in this outbreak investigation? What are the steps of contact tracing? Close contacts was household members, co-workers, caregivers, and those are caring exposed to infected animals or mosquito areas. The steps was to identify, list, classify, collect blood samples, monitor symptoms for 14 days and up to the final report. These pictures are some of the mechanisms which we use to trap or to catch the mosquito if we need to assess itself, like if it is having the disease. Okay, what are the biosafety and biosecurity measures to be applied to avoid the associated risk during
Sorry, may I see the first slide again?
You were agreeing to the pictures. The other slide, question D, it mentioned one is the CDC based and other is the CO2 based reference.
Actually, my question was, why you include the mosquito trapping as a contact tracing?
There is a question that. This is no sense.
Oh, okay. We move it to here. Yeah. It's here. Yeah, it's here. Okay. Yeah. Sorry. At that time, we were. Oh, sorry.
Yeah.
Never mind.
Okay. What are the biosafety measures that we should have to apply are wearing PPE, handling dead animals with care, disinfecting instruments, proper waste disposal, and disinfection of contaminated materials. Laboratory, it should be, or it should have to be using BCL3 for final cultural and molecular tests because of this is highly contagious virus. Also, we should have to use triple packaging. What are the observations expected by the survey team? Unusual level. These are the what observers have seen in the incident place. They have seen unusual death of pigs and horses or farm animals like cattle. Dispensing mosquito populations around the Lake Nasir and drainage areas, possible environmental contamination. What are the common methods of collecting various environmental and mosquito samples? Actually, the picture is from here. It belongs to this slide. It displays it to that one. I don't know the reason. Maybe he was afraid from here. The drain, okay, drain water, collect water from multiple sites for inspection. So when we are collecting the water, we should have to collect it different places. Last night when we were editing this, 1 liter was written here, and some of the participants said, why are we going to collect 1 liter? Are we going to use for a washing room to wash our body? Like that. So you need to collect it as much as water you need. You don't need to collect it like too much liters. Mosquito trapping. These are CDC Travis. CDC Travis is actually this one. Oh, sorry. And also oxygen-baited Travis, aspiratories, storage, transporting liquid nitrogen for virus isolation, PET sampling, live sera and carcasses tissue for post-mortem tests for further analysis, list the laboratory investigations that were applied for the diagnosis of causative agents and precaution during analysis. ELISA or maybe BCR. Also, if we are, since we are dealing first, we may need to isolate cell lines. Precautions include use of certified by safety labs PPE and decontaminations. What are the definitions of zoonotic disease and arbovirus? Zoonotic disease is infection naturally transmitted between vertebrate animals to humans, from animal to human. Arboviruses are arthropods viruses transmitted by blood, sucking insects such as mosquito or ticks. West Nile virus is a zoonotic disease, which is arbovirus and the family of parvovirus. What are the duties of mosquito, horses, and wild virus in the transmission of West Nile virus? Virus main reservoirs of an amplifier hostis, that is the main reservoir. Mosquito, they are the primary factors transmitting the virus from the reservoir to the to the human. Ecological correlation infection weak is when bird immigration and mosquito abundance coincide. Like there are, according to the climate change, some birds immigrate from one place to another place for some specific reasons. So maybe they may take the disease from one place to another, and the mosquito sac is spread from them, and maybe it can attack also somebody and transmit the disease like that. Do you think the Minister of Health managed this outbreak situation well? Explain your answer. This is a critical question. We cannot say no. Maybe where is Ali? Ali is not there. Maybe he can report to them and say those who are saying like that. No.
It's not valid.
Oh, okay. Yeah, that is the good news. Yeah, if he present here, maybe he could report to the Egypt government, and all of us will be hearing us. Maybe. Okay, we say yes, the minister, because of they did all these studies. So they did key management action is that action report responses have done is immediate information of a multidisciplinary investigation. They sent it the incident place to the team consisting of these specialists. Coordination has been established. Also communication, also public hotline, like they established call centers which serving for them, for the people. Sample are collected from human, animal, and factor samples are collected. International notification is needed, or they may have done it. Effectiveness limited geographical spread beyond upper HFT, mortality reduced to early identification and case management. High public compliance due to clear risk communication, and this is the conclusion. The minister's leadership demonstrated timely coordination, transparent communication, and effective vector control aligned with international public health standards. What are the risks? The risk communication element is considered during the disaster preparedness plan. Transparency should be considered, consistency, accessibility, community engagement, feedback loop. In the line with international health regulations, according to 2005, is it necessary to inform the WHO about outbreak? Even with international health regulatory criteria, this is they are saying we concluded that they, after assessing the disease has met all the criteria and it was serious public health impact and usual event risk of international spread, so notification to the WHO was mandatory. It should have to be informed. What are the recommendations to follow up at level is to resist this pandemic and ensure what it is that it does not reoccur in the future. Reoccur. Is to establish a One Health surveillance system connecting human, veterinary, and entomological network, strengthening the appropriate diagnostic capacity, regulatory and factor control, public health education campaigns before tourist seasons, intersectional coordination and stimulation, exercise for outbreak preparedness, maintaining stockpiles of PPE and diagnostic kits. Thank you so much.
Thank you very much.
Thank you so much. If you have some questions, you can ask, and also you can ask. But
And maybe we can pass the question to the part if you're agree.
I have a very small.
Yes.
Very simple question. Can you go to the question number 7 slides?
which slide?
Yes, question number 7
Number
slides.
4. Number 4. Then you are the next.
Like as you were mentioning that this was a case scenario in 2002, I guess.
Mhm.
So how can we use the WHO guideline of 2015 and 16 in 2002? Yeah,
You know, oh, okay, okay. I will answer. I will answer. Yeah. Look at you aware of every time. Yeah, that's good question. Let me try to answer. You will do the question. You know, every time there are publications and there are updating of, guidelines. So this guideline was existed because of triple packaging system is not something new because of the diseases. These diseases was not new. So it has been used. It has been using before, but they updated. So we took the updated version. Because
Maybe you can use these. I already put it here to explain about the categories. If you want, you can use the Biosafety Manual Guideline of WHO, the 4th edition, the last one. Actually, there is a, some sort of,
modifications.
classification for infections, disease, transportation. We will discuss. If you want, we can just take a scheme on it. Actually, in biological, we have 3 classifier of transportation: category A, category P, B, and exemption. And this is a fellow. If you answer all the question, you can find out where your sample actually belongs to, which category. In this case, we face samples that they are not cultured, that cause disease, and the disease are unknown for us. So it takes place in category B with UN number 33. And, you can see over here. And you can follow just the question. I will share this guideline with you as well for the precise answer. So thank you, group 3. Actually, maybe I think we can, ask a question at the end of participant presentation. Okay. Just wrote it somewhere to remember.
Hello, Assalamu alaikum. My name is Navira and I'm the member of group number 4. And the members include the Navira, Mariam, Rabia, Sumbul, Hasifa, and Khadija. Okay, so the outline of today's presentation is that this case study is divided into 5 major parts. One is the nature of the case study. Light versus lights. What's wrong? Light says laser. Okay, So nature of the case study, settings and background, nature of outbreak, epidemiological findings, trainings and learning objectives. So nature of the case study is that engaging the training scenario for realistic public health application. So this is basically a template. If you have any outbreak, so you can use this kind of template for your investigation. So engaging training scenario, interactive learning via simulated outbreak investigations and response. Settings and background. So the scenario they have given, it's a fictional scenario that represents the outbreak occur in the Aswan, Upper Egypt in 2002, chosen for climate ecology supporting disease transmission. The scenario uses real research data from Egypt to improve realism and context. So sudden severe febrile illness of the unknown origin with a high case fatality rate, neurological symptoms and intestinal disturbance are predominant clinical manifestations. No clear etiology exists leading to extensive investigation. So most of the cases linked to the direct contact with the farm animals, especially equines and birds. Equines means donkeys and farm cattle. Additional cases among those near waterways, including tourism staff and tourists. Infections and deaths primarily in the Aswan, limited spread to the neighboring areas. So in the training and learning objective, it's step-by-step outbreak investigation, case detection, hypothesis testing and containment, epidemiological challenges with emerging pathogens, importance of biosafety and biosecurity in response, skills in risk assessment, coordination and data interpretation. So scenario, so I think we all have been through the scenario number 1. So the question of that scenario, that was just basically the introduction of Aswan, and they are describing in that scenario that how it is connecting with the different regions. So the question number 1 is, what are the factors that make Aswan governorate an interesting area for epidemiological investigation? So what we have searched and what we have found, that zoonotic risk, tourism-related exposure, water-associated hazards, climatic conditions, cross-border connectivity, rural and agricultural lifestyle, and environmental contamination. So zoonotic means close contact exists between human, farm animals, and wildlife. Tourist animal contact increases pathogen transmission risk locally. Nile proximity aids vector breeding and waterborne disease spread. Hot, dry climate benefits the vectors and pathogen survival. Egypt-Sudan link aid the movement, risking cross-border disease spread. Rural population farming, increasing zoonotic exposure risk. And wastewater disposal risk, contamination, disease. So now the question number 2, design a case investigation. Data from form to follow up on the onset of the disease exposure related to the information, time of sample taken, and show details. So we have generated a form. So this kind of form can be prepared for sudden outbreaks. So the question number 3.
Can we just skim the main character? Okay, so then.
Okay.
That's okay.
This is half part.
Okay, so then.
Mm.
Okay, just skim. And then see that the other part.
So in A part, case identification, case ID, patient name, initials, and age, sex, address, occupation. Then clinical information, date of symptoms of onset, date of first healthcare visit, symptoms at onset, maximum record temperature, neurological symptoms, and so GI symptoms, outcome, and date of outcome. Then in C section, you have exposure history 14 days before illness, exposure type, that includes the direct contact with animals, contact with blood tissues and excreta of animals, insect or mosquito bite, work or stay near the water bodies like Lake Nasser, Nile, and drains, recent travel within or outside the Aswan, contact with similar cases, consumption of raw or uncooked, undercooked animal products. Then in D section, laboratory samples information, you can add the sections like sample type, date taken, time sent and received to hospital, lab code IDs, and remarks. Then epidemiological linkages include ID, relationship contact, last date of contacts, shared exposure, and laboratory sample relation. Then in F section, you can include the investigator name, designation, organization, like who the person who is taking all the details and investigation. So what you can do, you can label the samples with patient ID, date, type prior to sending, verify the epidemiological connections among the cases, conduct daily update, clinical status, laboratory findings, document extra suspect cases for uniformity. So question number 3, what are the clinical and post-mortem investigation that are applied to the diagnosis and cause of the cases? So in clinical pathology diagnosis, you can take the samples from the blood, CSF, fluid samples, stool samples, endoscopy, MRI, and serological analysis. CSF samples are difficult. So initially you can take the like initial request samples can be the blood samples, stools, and urine samples, or MRI you can request. And later on, if you find anything abnormal, then you can request for the CSF, because CSF sample collection is very difficult and it is only requested on the very urgent or very required cases. In post-mortem investigation, tissue histopathology, liver, lung, brain, or for pathogen detection. I think brain would be difficult, but whatever the infected people who are not recovering, who are died, so from them, I think these samples can be taken. So what are the recommended samples to collect from the patients according to the mentioned symptoms? So these include the patients presented with the fever, neurological symptoms, seizures, encephalitis, GI disturbance, rashes, and a wide range of biological specimens should be collected, like blood sample, CSF, tissue, urine, stool, and swabs. So this was the another case which was a scenario that was described, and the question was describe and classify the different suspected causes that may lead to the febrile illness symptoms of these cases. So that include the viral causes, bacterial causes, and parasitic causes. In viral causes, West Nile virus; in bacterial, Leptospira species; and in parasitic, Plasmodium. So you can check for these organisms. What are the possible ways of transmission of this unknown disease? So the suspected disease can be transmitted through following routes: vector-borne transmission via bites of infected mosquitoes, zoonotic cycle, bird to mosquitoes, mosquitoes to human or horses, or human and horses are dead and host. Other rare routes are blood transfusion or organ transplantation from infected donors, vertical transmission from mother to fetus, or breastfeeding, laboratory exposure, or needle stick injuries during sample collection. Okay, so this was the another scenario that was given, and it corresponds to the question number 7. What type of packaging should be used to transport the samples and why? So as we've already discussed in our previous lectures, that we need to, in such scenarios, we need to use the triple packaging, seal proof containers. Second, you have to collect the blood samples, then you have to put those samples in any additional packaging material, like you can put them in the 50 ml Falcon or and so on, and then you have to put them in the bags and then containers. So samples will be transported in triple packaging because the agent has not been identified yet, causing variety of the symptoms, including respiratory failure. So it would be considered as a BSL level 3 risk. Which type of surveillance technique do you think must be activated and conducted during approach of the case, and why? So one is the syndromic surveillance, other is laboratory-based surveillance, and third one is sentinel surveillance. So in syndromic surveillance, monitoring the symptoms and pattern of illness of affected individuals. In laboratory-based surveillance, tracking laboratory confirmed cases. And in sentinel surveillance, monitoring a specific population or sites for the early warning signs such as horse riders and tourists. Do you consider this disease outbreak as severe? How do you define this and why? So, severe outbreak with high morbidity and mortality rates is a high fatality rate is 10%, and rapid spread among all age groups and tourists. Neurological complications signify serious pathogenic impact. Emergency declaration necessary. Public health and emergency declaration. Okay, so question number 10, review and complete this sheet and showing all the details of the recorded cases and follow that what has been done. So here we have to, this is the chart or table that is given, and we have to complete this by the missing information such as total recorded cases. So there were total cases 130, of which males were 73 and females were 57. Recovered cases were 114, and among which the 66 were males and 48 were females. Death rate was very low, like 16 of the total cases died. So draw an epidemic curve and according to its shape, try to assume the pattern of infection. And as you can see that in blue here, there are the new number of cases, and in orange, there are cumulative deaths. So cases are more comparatively to the deaths. So epidemic curve indicates the propagated spread via mosquito vectors, helps define exposure period and evaluate control effectiveness. Identify who are close contacts and what is their role in this outbreak investigation. What are the steps of the contact tracing? So close contacts people are the people who are near the west, who are in the Aswan, and they are affecting with the West Nile virus. Surveillance can be tracked by the secondary cases, and public health effort halted the transmission. Then community engagement tackle febrile illness. Data can be traced down from the identified virus sources, and collaboration strengthened health responses, and rapid response teams enable the investigation, and monitoring systems of the healthcare should be used for the lessons for the future strategies. What are the biosecurity measures to be applied to avoid associated risks during their work inside the laboratory or outside? So for the biosafety, you must have the personal protective equipment, gloves, mask, BSL 2 and 3, waste disinfection, sample labeling, and restricted access for the lab staff. What are the observations expected by the survey teams? So many mosquito breeding sites near the drain ponds and Lake Nasser. Dead wild birds, especially crows, some dead horse farm animals, mosquito swarms observed around the animal areas and stagnant waters. Human cases mostly from those living or working near the water or farm sites. What are the common methods for collecting various environmental and mosquito samples? So, adult mosquitoes, CDC, light traps, aspiration, or gravity traps. Larvae can be collected by the water dipping from the stagnant pools. Bird samples can be collected blood for testing. Water samples, 1 liter from each drain of ponds and drains for larva examination. Keep all samples cold and properly labeled. So list the laboratory investigation that were applied for diagnosis of the causative agent and precautions during analysis. So for test, you can do the ELISA, detects the antibodies, then you can perform the RT-PCR, detects viral RNA, IFA, virus identifications in the cultures. Precautions, use BSL level 2 and 3 lab safety, wear PPE, avoid aerosol generation, triple packaging and disinfection of the working area. List laboratory investigation that were applied for the diagnosis of the causative agent and precautions during analysis. So zoonotic disease, infection that pass naturally between animals and human, is spread through direct contact, bites, vectors like mosquito. Example, rabies, brucellosis, West Nile virus. Arbovirus, arthropod-borne viruses spread by mosquitoes, ticks, and sandflies. Multiply in both vector and animal-human host. West Nile, dengue, Zika, and chikungunya. Okay, what are the roles of the mosquito, horses, and wild birds in the transmission of the West Nile virus? So, S1 outbreak linked to the West Nile virus, a zoonotic flavivirus. Culex mosquitoes identified as primary vectors. Mosquitoes acquired the virus from viremic birds, maintaining it for life. High mosquito density near Nile water facilitated transmission. Bird, especially migratory species, serves as amplifying host. Horses and donkeys act as sentinel hosts for the viral activity. Human and dead and host indicating environmental transmission pathways. Transmission cycle is enhanced by climatic conditions and high densities. So question number 19, do you think the Minister of Health managed this outbreak situation well? Explain your answer. So timely actions by the Minister of Health controlled the outbreak effectively. Rapid response teams were sent to Aswan Hospital, reported unknown fever cases. Enhanced diagnostic confirmed West Nile virus. Insecticide is sprayed, reduced mosquito populations. Insectorial coordination supported rapid surveillance. Public communication mitigated fear and misinformation. Outbreak curve declined sharply. No new deaths post December 11. Minister of Health response is effective under resource limitations.
So your answer is yes.
Yes. So, yeah. So, question 21, in line with the international health regulation, is it necessary to inform the WHO about this outbreak? So, obviously, it is essential and necessary to inform about any outbreak happening. So, as one outbreak warrants reporting to the WHO under IHR 2005, cross-border relevance due to proximity to Sudan and high human-animal movement. Risk of international vector-borne infection is spread increase. Infected tourists could lead to exported cases in the Europe meeting IHR criteria. West Nile virus is a zoonotic arbovirus with potential to re-emerge globally. 16 deaths and neuroinvasive disease classify it as a PHEIC by WHO. Reporting enhances international collaboration and compliance with the IHR, improving global arboviral surveillance. So, what are the recommendations to follow at all levels to resist this pandemic and ensure that it does not reoccur in the future? So, implement comprehensive One Health-based strategy to prevent the outbreaks. Strengthen integrated surveillance linking human, animal, and vector monitoring. establish continuous mosquito controls programs with larval sources management and insecticide spraying, enhance regional laboratory diagnostic capacity with ELISA and RT-PCR, sustain public education on mosquito repellents and early medical consultation, include veterinary surveillance for birds and equines as viral activity indicators, foster intersectorial collaboration among health, agriculture, environment, and tourism, invest in research workforce training for vector ecology and biosafety to increase the resilience. So, conclusion, S1 2002 outbreak study applied epidemiological principles to a West Nile virus case, focusing on outbreak response component. Investigators examined the environmental factors such as climate, Nile proximity, migratory birds affecting the vector-borne disease, highlighted importance of early identification, laboratory surveillance, and vector control to reduce the health impact, emphasized risk communication and international cooperation under IHR for global health events, demonstrated One Health approach integrating human and environmental health for disease control, served as a training model for epidemic preparedness and critical public health action. Thank you.
Great, thank you. Thank you very much. Maybe we just past 5 minutes, maybe empty anything and go on with group number 1. No, where the last one? You asked to be the last one? Maybe. Okay, we will continue with group number 2 and then then share yourself. No, it's. Yeah.
What's it? आपको देने सेंटर में जिसके एक मिनट जैसे हैंडिंग आउट विशिंग सर्विस इसमें यूएसबी स्लॉट ही नहीं दोस्तों नहीं पॉज कर दो स्टॉप
नहीं
Assalamu alaikum everyone and very good morning. Myself Savira Rajput, and along with my group members, I am going to present a case study that is on emergence of febrile illness of unknown cause among the population and visitor of Upper Egypt.
And you can use that mic as well. Put it in your bag and move wherever you want. Okay. And pin it on. Okay.
Let me start again. Okay. So let me introduce myself. So my name is Savira Rajput here, and along with my group members, I am going to present the case study that is on emergence of febrile illness of unknown cause among the population and visitor of Upper Egypt. So this is basically a case study. For this, we have different scenarios. During our conversation between group, we conclude some idea related to question. So according to the first question that we have, what are the factors that make S1 governorate an interesting area for epidemiological investigation? So for this question, first we have to focus on this graph because the geographical location for this is very important for epidemiological importance. So the S1 is basically located in Upper Egypt, bordered by two different countries like Sudan, Red Sea in the east, and several governorate are, we can say that they are interconnected with each other. So we are focusing on what type of infections or outbreak occur in these different countries. So we try to find out on Google that in Sudan, mostly West Nile virus, dengue, malaria, and especially bacterial meningitis that cause neurological symptoms. That is discussed in earlier questions. So in next on the Egypt, the Cairo outbreak route various sewerage waters, West Nile viruses in 2002. This will be the suspected infection could be. So other epidemic importance could be climate. There's the S1 city has a very hot climate with the harsh environment. Of course, water source will be one of the important key factors. And last but not the least, the tourism and the human movement. So this will cause the vector-borne diseases that can be easily transmitted to one border to another border. So the second question in which we have to make on the basis of onset of disease, exposure-related information, and sample collection time, we have to make an investigative case scenario and make a form for this. So we had made this. For this, in first section, we have a demographic information related to a patient in which age, sex, gender all are included, like patient history as well. Secondly, what will be the clinical symptom that the patient has suffering from? We have to mention all these accurately here. The third one is the exposure information. And fourth one is specimen information, like what type of sample we collected from that suspected person. And finally, the timeline summary. So when will, how much time as a patient is suffering from this type of disease as well. So finally, these type of data basically help us to link between how much time disease will take progression and how to link with different symptoms and correlate the final result with the lab to another doctor or specify our results. And the third question related to scenario is that what will be the clinical and post-mortem investigation that are applied to diagnose the cause of different cases. For this, we have to first read the below scenario that is given in the case study. So first, the clinical. The clinical one could be MRI detect brain inflammation, ELISA identify antibodies, RT-PCRs confirm the viral RNA, CBCs and liver enzyme axis systematic impacts. And for the post-mortem, histopathological of brain, liver, lungs, and GI reveal tissue damage, virus isolation, PCR confirm the pathogen presence. So, for the question number 4, what are the recommended sample to collect from patient according to the mentioned symptoms? So, we have to select what type of sample. So, we have to collect data from this scenario as well. So, sample could be a blood. For serological tests like IgM, IgG, PCR, or for biochemical test. And for CSF, like patient of essentially the patient with seizure and expert, like this is related to the brain. And tissues from disease case of histopathological and molecular testing. We can take skin swab as well, like if a person is suffering from in this, like as you see in this picture, like person is suffering from some sort of skin rashes. And finally, the environmental sample could be like mosquitoes, birds, and water samples to trace various vectors and reservoirs as well. Question 5, describe and classify the different suspected causes that may lead to the febrile illness symptom of these cases. So basically, in first scenario, we will observe that a person is suffering from different symptoms, but the medication that is provided to the person is not applicable. The person is still having a fever. For this suspected case, we have to find out what will the possible causes of the disease. So according to our suspicion, it could be first viral, like it would be West Nile virus because some, according to some literature review or some information that we find in given scenarios, we make a correlation between them. Dengue, chikungunya, like these all are vector-borne diseases. The vector is mosquito. And second one could be bacterial like typhoid, leptospirocopy linked to a water and animal resources. It could be parasitic because symptoms also related to parasite like malaria, endemic in the border is connected to like Aswan city is also connected to the Nile River. So we can say that from Nile region, it could be a parasitic like from water and environmental. Uh, uh, as we assume that the Aswan has been facing a very harsh environment, hot climatic changes there. So it could be a chemical changes, or we can say the exposure of different chemicals as well. That is a consequence from these exposure as well. And finally, it could be a zoonotic, like direct contact with infected birds or any others. Question 6. So what were the possible ways of transmission of the unknown disease? So for the disease that we are investigating, we will find out that as it would be a vector-borne, it could be zoonotic, environmental, or rare to human transmission as well. So the question in this case study as well, like what type of packaging should be used to transport the sample and why? I think this question is very important in the scenario if the sample is viral, bacterial, live, or dead, how we transport from one area to another area if we have laboratories in distinct regions. So first we have to use triple packaging system as we already discussed in different lectures and different safety guidelines as well. So the primary container, it could be a leak-proof tube with a label sample, obviously. And secondary container, it would be a watertight box. It might be, a sample could be a virus, a deadly virus that is unknown. It could be cause of outbreak as well. So we have to tightly pack in a container. It could be a vial with the absorbent material. If the leakage is occurred, so it will absorb by absorbent material. And finally, outer container as well. So we have to place our primary and secondary containers in the third container. That could be rigid, labeled, or biohazard symbol bag. It could be a box or a thermocol box. It could be to protect the handler, consumer safety, integrity, and compile with the WHO, WHO. And if we want to send our sample with the help of airlines, so we have to read the standards of IATA. Okay, now the final, next I want to call Mr. Danish. Please come on the stage and continue our presentation. Thank you.
Assalamu alaikum, my dear colleagues. Myself, Danish Hassan, and I'm here to present and continue the following questions. Thank you, Savera, for presenting and explaining the previous scenario. I'm here to present from the question number 8. Which type of surveillance technique do you think must be activated and conducted during the approach of the case, and why? After reading all the scenario, we pick the surveillance after reading the scenario. These technique will be applied after the outbreak or before the outbreak. The active surveillance field teams identify case proactively, and the second one is syndromic surveillance, track cluster of symptom before the lab confirmation. And the third one is vector surveillance, monitor mosquito species and infectious rate. Zoonotic surveillance, detect disease in the animal to prevent spillover to the human. Why we choose choose all of these? Because after the active surveillance, there are active cases rise in the swan. That's why we choose active surveillance. After the syndrome, we choose syndromic surveillance. There are required 5 to 7 days for the testing histopathology sample. That's why we take the syndromic surveillance. We can take a cluster of symptoms in syndromic surveillance. And I'm telling about the vector and genetic surveillance. After the history of the patients in the Swan and Cairo and Kenya, they are contact with mosquito bite and also with animal. That's why we took these two. Now moving to question number 9. Do you consider these disease breakout as a severe? How do you define and why?
Actually, I mean, it's better to. No, it has a lot of noise and put it in your bag, the debate. And put it in your pocket.
Thank you, ma'am. In our opinion, after reading the scenario, yes, we are agree with the severity of the outbreak. How we define is because of the high case fatality rate. After there are 130 cases is were recorded at that time, and 17 were death. Then the case fatality rate is high, and it's about 12%. And especially the death will include the children and elder. And the second one is neurological complications, and the third one is rapid spread across the gonorrhoeae. It was not the localized outbreak. It moved toward from the Aswan to the Cairo and also Kenya. And the fourth was also international exposure. The tourists were came to the Aswan. That's why international tourists also affected from the outbreak. This is the overview that's showing all the detail of recorded cases. Look at this. The total cases is recorded 130, and in 130 there are 73 were male and 57 were female. And after that, 114 were recovered. 114 were recovered. In 130 cases, there are 105 from Aswan and 25 cases from the other places. 15 from the Kenya and 10 cases from the Cairo. And 114 out of 130 were recovered, in which 66 were male and 48 were female. The case fatality rate after the death, 16 out of 130 is 12%, and in which 7% is male and 7 were male and 9 were female. That's all the overview of the cases were recorded. Okay, thank you for listening me. After this, Rizwan will continue from the question number 11. Thank you. Zaza.
Bismillahirrahmanirrahim. Thank you, Danish. This is me, Rizwanullah. Now I will proceed the further presentation. This is the epidemic curve of the outbreak, how it will indicate. The epidemic curve indicate as that during the period of the time, how many cases occurred, whether how this cases occurred. This is the chart of epidemic curve. Here are some orange line and the blue chart. The orange line shows the death cases, how many death cases are happened, and the blue chart shows the recovery cases. Here are the horizontal rows shows as the death in the period of the time that cases happened, and the vertical shows as the number of cases happened. For example, in this period of time from 4 December till to the 6th December, there are few cases of the recovery cases and also the death cases. It means this phase is index phase or incubation period of the West Nile viruses. As we know, the West Nile virus incubation time is 2 to 14 days. In this incubation time, there are no symptoms, no clinical symptoms. We don't know which kind of disease is this. This phase is called index phase or incubation phase. After that, dramatically or drastically increased happened. There are lots of death and recovery cases happen. It means this is intense or high transmission period of the outbreak. Here is the intense or high transmission. It means in this from 7 to 11 December, it is the peak time of the outbreak. In many cases happen and many transmission occurred. After the 7 to 18 December, it is the control or end phase because there are no death occur, but the recovery phase become more increased. For example, in this phase, we know what is the disease, what are the viruses, and how can we control these viruses. We have all the information about the outbreak, and then we control the outbreak. Okay, thank you. Okay, the question is like, how can we identify the close contacts or what are the rules in the outbreak? For example, first of all, before answering this question, we should know how the West Nile virus is transmitted. The West Nile virus is zoonotic or maybe it can vector-borne disease. The human to human transmission is very rare and it cannot be transmitted because in the human blood, the load of the virus is so few. The mosquito bites the human, then human can be infected, otherwise cannot be infected. That's why, due to this reason, we can observe the family members. For example, we observe the family members, we observe the neighbors, we see how it happens from the local mosquito or from the maybe from another area. And also we can observe the co-workers, or we can see the close contacts of co-workers or lab staffs, because lab staffs are doing the test and they are very they are in high range of dangers that they can expose to the virus. Also, it is zoonotic virus or zoonotic. It can cause the zoonotic disease. We can also examine or observe the animal, the people who are handling the animals, like within an area where the farmers are doing work on these things. And also, as we mentioned before, there were some tourists come to the Aswan, and they were infected here, and they bring some diseases or some viruses with yourself. We can also examine this. What are their rules in the outbreak investigations? Their rules are that we can identify the exposure site of this virus, how it exposed. For example, it is infected, these people are from work site, from home, or from any other place. Due to that, we can examine the signs, symptoms, and then we can control it. And what are the steps of the outbreak examination, or what are the contact tracing? For example, first of all, we confirm that is confirm the disease, which virus is that. We did all the laboratory tests. In this, the confirmed case come with us. Then what will we do? We do the interviews from the patient who are already infected. We will ask them, okay, did you travel in area or not? What is your before 2 to 14 days? Did you have the symptoms or these things? You will ask these questions. After the interview, what will we do? We will have the signs and symptoms and confirm patient. We will monitor or control those symptoms, and we will collect the samples from different area, from humans, from the environmental places, or from mosquitoes or these things. Okay, go ahead. Okay, question number 13 is, what are the biosafety and biosecurity measures to be applied and avoid the associated risk during either inside the laboratory or outside the laboratory. As we know that West Nile virus is dangerous to us because it can cause encephalitis or also meningitis, which is very severe condition. We should be careful while we handling these viruses. It can be so dangerous to us because we should use the proper PP, mask, gowns, and also disinfect the area where we are working and dispose the best material. And we can work in the B cell 2 and B cell 3. As everyone mentioned, we work in the biosafety level 3 and biosafety level 2. But which viruses we conduct in biosafety level 2, in which viruses we conduct in biosafety level 3. Inactivated viruses we conduct in biosafety level 2, and activated or live viruses we conduct in biosafety level 3. Because inactivated, what does it mean inactivated viruses? Whenever we are using formalin or some chemicals in the samples and we inactivate that virus, that becomes inactivated virus, and we can use those viruses for formation the vaccines and other, like we are dealing CSF samples and blood samples directly. First of all, we inactivated the virus, then we deal in biosafety level 3 and by 2, sorry. And biosafety level 3, we examine the animals or those mosquitoes that are infected. Okay. And
Points are on Kazar Kawai.
and what are the observation or expected survey teams do? The survey observation are done first of all by humans. They are just collecting their data, how many expected cases happened and what are the confirmed cases. Then we can also observe the horses or mosquitoes, and we can also observe the poor water sanitation of these things. And what are the methods of common collecting various environmental and mosquito samples? There are some methods which can be used for collecting those samples. One is CDC live traps. CDC stands for Control Disease Center. They use a device which means live traps. They use a light which traps the mosquitoes or these things. Then we can examine those mosquitoes. Aspirators, they use a device like aspirators that dip in the water and aspirate the water, and then we further confirm whether it is West Nile virus in those waters or not. And also we can use blood or swabs from GIT tract, like it is a part of GIT cloacal swabs used. And also we can use the sample of water. Thank you so much. It was my part, and further parts will be presented by. Thank you.
It will watch the time as well. We will just have 10 minutes.
ये बस जल्दी जल्दी खत्म कर ना।
Okay, am I audible?
16.
Okay, let's now continue from the question number 16. List the laboratory investigation that were applied for the diagnosis of the causative agent and precautions during analysis. So we have to test the serological testing, molecular test, histopathology, and virus isolation for the detection of the, as we consider it as the West Nile viruses. Serological testing, including the ELISA test, plaque reduction, neutralization test for the confirmation, molecular testing, including the RT-PCR, histopathology of the brain and liver tissues. The precautions including handle all samples under the BSL-3 precautions, use sealed centrifuge and biosafety cabinets, decontaminate instruments and surface immediately after the use. Okay,
For the question number 17, what are the definition of the zoonotic diseases and arbovirus? As we already know that the zoonotic diseases is the infection that is transmitted between the animals and the humans, including the rabies virus or the vaccinal. Also, arbovirus virus is transmitted by the arthropods, including the mosquito or ticks, etc. Example: vaccinal viruses, dengue, chikungunya. We are very familiar about it. Question number 18, what are the roles of the mosquito, horses and wild bird in the transmission of vaccinal viruses? Okay, first of all, we know that the mosquito is a primary vector. Species a closest species. That wild bird is the key reservoir. It amplifies the viruses and basically replicate it. Horse is a dead end host, show clinical signs but do not transmit it further because it is a dead end. As we know, as we here mentioned that it is the dead end host. Question number 19. From the case studies, do you think the Minister of Health managed the outbreak situations well? Explain your answer. From the previous we have discussed in the group, we conclude that yes, the Minister of Health responses very well because it immediately effectively immediate activation of multi-sector responses, including the health, agriculture, and environmental sectors. Initiate public communication via media and hotline. I will also mention the hotline number. Ensure laboratory confirmation and transparent reported. Implement biosafety and vector control measurement. Vector control measurement is very, very important. Question number 20. What are the risk communication elements considered during the disaster and preparedness plan? The risk communication basic elements including the timeline, information sharing on the daily basis reported, transparency with the clear communication to the government and the public also about the risk and prevention actions, community engagement, inform the public on the mosquito control and the personal protections, media collaboration to prevent misinformation. Feedback mechanism. This is the hotline number that the Osman government used for the symptoms reporting for future cases. Trust building, consistent updates to maintain public confidence. And we have to train our public and the staff is also. On the question number 21, in line with the, what do you think the international health regulation, it is necessary to inform the WHO about this outbreak? We conclude that yes, because it is mandatory to inform that World Health Organization under the International Health Regulation 2005, because this, as we conclude that this outbreak involves a zoonotic or arbovirus potential invasion, or it meets the criteria of Index 2. Serious public health impact, unusual events, and risk for the International Travel Association transmission. This notification allows global coordination, support, and resource mobilization. For the last question, question number 22, what are the recommendations to follow the level of the resist of the pandemic in the future does not recur? Okay, it will be applicable for all level, on the local and public level, national level, and international on the WHO level. Local and public level, it is very, very important for the personal protections. Environmental control, remove stagnant water because it is a vector-borne diseases. Community reporting, notify authorized person about any suspected cases. National government level, from the national or government level, surveillance must be done on the daily basis. Rapid response active trained team for the outbreak investigation. Vector control is very important. Health preparedness, upgrade labs, and train healthcare staff. On the level of international and WHO, global tracking, monitor reports of the outbreaking internationally. Communication is very important. Share timely alert and updates. Okay, this will all about our group. Hope you all enjoyed and like our presentation, as we do our best. If not, we will be do our very best for the very next time, inshallah. Thank you.
We have another group after Group number one, the NID group, they just create the scenario tomorrow, today morning, and in limited time, they prepare to have a presentation. So after this group, we have another one, and then we discuss together. Um,
Okay. Why do I zoom it? Zoom it? This. I need to zoom it. Let it be big. Yeah.
You cross the bookmark. Here,
Okay.
then here.
And then
It's okay.
it's okay.
So
Yeah. Okay.
you can avoid this.
All right. Thank you. Good morning, everyone. So my name is Ekanya. And I'm going to be doing a presentation for group one, which is made up of these fine faces. We have Amna, myself, Sadaf, Sophia, and Rashid, the lone male in the group. So we are like the rapid response team doing an investigation on the emergence of febrile illness of unknown causes among the population and visitors of Aswan, which is in Upper Egypt. So our objectives, our learning objectives was, first of all, to understand the dynamic of the natural history, to distinguish the classification and biological characteristics, and then demonstrate the association of geographical location with disease, and then to explain the role of different host sites for spreading diseases and discuss proper collection of samples, packaging, transportation. Okay. As well as apply these strategies for the prevention and control of a newly emergent infectious disease like the one that we are dealing with. At the end of the day, we're going to demonstrate suitable schedule for treatment, protection, vaccination if available, and make recommendations to stop future outbreaks. So our study area, it's in Aswan. Aswan is, okay, Aswan is what we have. Border to the south by Sudan, to the east with the Red Sea, and with other governorate areas as well in Upper Egypt. So we have our location there. So you can see that's our Aswan location with the Red Sea. So this is the case we are dealing with. It's a fictional case, but since all of us already know the case we are dealing with, we are just going to go straight to responding to the questions. With our very first question, that says, what are the factors that affect us, that makes us an interesting area for epidemiological investigations? For epidemiological investigations, we have to, what makes us very interesting is the fact that it first of all has records of past history of outbreaks like Rift Valley, West Nile in different years. Also, it is a unique environment with different ecological conditions. We have forest areas, we have zoonotic disease, we have coastal areas. We also have urban slums that makes it suitable for mosquitoes to breed. We also have the population characteristics that have different occupations. We have farmers as well as tourist guides that makes them very close to wildlife and propagate or be at the risk or front line of disease contact. And then our second question says we should design a case investigation data form to follow up the onset of the disease. There is actually a standard case investigation data form by WHO, and this is how the standard case investigation data form looks like, where it has the general information about the patient. It has the clinical information about the patient. It also has an exposure and epidemiological information. It also gives you a laboratory investigation information, like what samples you collected, what results did you get? And then it also has a follow-up and of course tracking, because most times in case of outbreak, if within the local area, if you can't, the area doesn't have the facility to do confirmed tests, we have to transport the samples to other places. So tracking the sample is very important. And then we also have the place where the investigator, who is probably the health authority, has to sign. Here now we have our field case investigation form, where we have all the patient information, the clinical information, according to what was given to us in the case study. So we have the information, exposure and travel information of our patient, as well as the laboratory information. Question number 3 says, what are the clinical and post-mortem investigations that are applied to diagnose the cause of the case? Of course, we know what clinical investigations are. Clinical investigations, we need to collect clinical samples and then we run tests. In this case, it was important we collected blood samples. The blood samples was for us to do hematology tests, biochemistry analysis, to actually check for elevated enzymes or bilirubin cases, or elevated bilirubin that are very often common with Rift Valley or other viral infections, considering that we don't yet know the disease we are dealing with. We could also use the same blood to do serology analysis to actually confirm if it's a recent infection where we can get the presence of specific antibodies to Rift Valley, West Nile, and other viruses. We could also as well do molecular tests, which are very confirmative, or we could do microbiology analysis to do culture. And we also collected cerebrospinal fluid to actually, it helps confirm the presence of viral encephalitis, which is very common with West Nile infections, considering the fact that at this point, we don't yet know what disease we are dealing with, okay? And then, with the samples we got, we could also do imaging, that's brain imaging as well. For postmortem investigation, postmortem is for somebody who is already dead. So for postmortem investigation, we collect biopsy, tissue biopsy from different vital organs, and then we do histopathology. For our question number 4, it says, what are the recommended samples to collect from patients according to the mentioned symptoms? Based on our symptoms, we're able to say that we collect blood samples. You know, blood samples could give us basically most of the information that we'll need. We could collect CSF, as well as we could collect sputum, with possibility of, if there is since it's a viral infection, it maybe, just maybe, it could be transmitted by droplets, just maybe, and things like that. And then for post-mortem case, the samples we collect are blood samples, or we collect CSF, or we collect biopsy, okay, actually, we actually differentiated this because at this point we had 3 cases, 2 brothers and a woman who actually reported death just after being brought to the hospital. So our next question is, we should describe and classify the different suspected causes that may lead to febrile illness symptoms of these cases. Since we don't yet know what possible infection, there is a possibility that it could be arbovirus infections. There is a possibility that it could be other viruses. There is a possibility that it could be bacteria of zoonotic origin, like brucellosis and other diseases like that. There is also a possibility that it could be parasitic disease like malaria. And we also have toxins from chemical exposure. So there are a lot of possibilities at this level, since we don't yet have any idea of what we are dealing with. And then what are the possible ways of transmission? Based on the implication of the vector, based on the implication of animals, based on the implication of environmental exposure, based on the implication of humans, we could say possible transmission of the disease would be vector-borne transmission, okay? Zoonotic, which is animal to human. We could also have environmental exposure. We could also have a rare situation of blood transmission. We could also have a person-to-person transmission. But we are aware that when it comes to viral diseases like West Nile, when it comes to zoonotic arboviral diseases, person-to-person transmission is rare. But there is a possibility. And this led us to the next question. Now, what type of packaging should be used? Of course, from this situation, or how the deaths are being recorded, or how cases are being recorded, we could already know that it's a severe situation. So we'll need a triple packaging system of transporting our samples. And which type of surveillance technique do you think must be activated and conducted during this kind of approach? Surveillance here, looking at the intensity of the outbreak, we chose to go with a combined surveillance system. A combined surveillance system where we are going to be using event-based based on the situation at hand. We could use case-based, that's where we detect the cases, or we go with active-based. Active-based surveillance means we will not only wait in the hospitals for cases to be reported, we move to the communities to get cases as well, do sampling, do testing, do investigation to get cases, as well as sentinel, where we have to detect infection in the animals and in the vectors. There is also laboratory-based, where we have to confirm diagnosis from the samples that we have. So it's a combined surveillance system. One method alone cannot be enough to survey for this kind of outbreak, okay? And then, question number 9 says, do you consider this disease outbreak as severe? From where we are coming from, it's very clear that it's severe. And then if you look at the assessment metrics, which has to do with likelihood and consequence. If you put the likelihood, there is obviously a high likelihood here, and the consequences are severe. So we are going with the fact that yes, the outbreak is severe, because it involves high number of cases. It shows rapid spread within the population. It shows serious clinical outcomes, and it shows a significant social or economic disruption. So we agree that it is a severe outbreak that we are dealing with. Question 10 says we should review and complete this sheet showing the details of recorded cases and follow what has been done. So we have our summary down there. We had altogether from everything, 130 cases, 73 which were males and 57 females. Amongst them, 114 recovered. Okay, so there's a high recovery rate. 114 recovered. And what we discovered about the disease is that, okay, it's a disease that treating just the symptoms alone will make the disease go away after some days. At this point, that's what we could discover. And then we also could notice that age population, who are very young, between 5 and 12 years, they have the possibility of dying from the disease. Then another age group of above 60 years also have a high rate of mortality from the disease. But the middle age has a higher chances of recovering from the disease. So that is what would start leading us or giving us information of what kind of disease we are dealing with. Okay. So we had to draw an epidemic curve and according to its shape, try to assume the pattern of the infection spread. So we have with us an epidemic curve, which is drawn by hand, especially by Amna. Interesting one. Okay, so this is a curve of timeline of illness on the x-axis and number of cases reported on the y-axis. So by virtue of the shape, we see that cases, we started recording cases from the 4th right up to the 18th to or 20th or 20th of the month. And we have between the 4th right up to the 12th, we have high number of cases recorded. And then after that, the cases actually dropped. Though cases were recorded in other sites, not as intense as what we got from the end to tell you that there's actually a possibility of the plan that was used by the response team actually was working very well to curb cases as fast as possible within 1 month of the outbreak. And then identify close contacts and what is their role. Close contact in this case, we have tourists, of course, because the disease, if it is a tourist guide that was reported with a disease, then he has been moving with tourists. So there's a possibility that our tourists from other countries could have been, they have been in close contact with this patient. We have the family members of our patients. We have students, considering that we have 2 students from a school. So if they are coming from a school, it means the whole school probably was in contact with these children. So they too are at risk of this disease. Of course, the health professionals and farmer. What then are the steps that we use to do contact tracing? Why do we first of all do contact tracing? We do contact tracing to actually stop the chain of the disease, okay? Because if we are fighting and concentrating only on those who are already infected, there is a possibility that others who are not yet infected could be getting infected. So we quickly do a contact tracing to actually support, manage situation so that we break the chain of any further disease transmission. So that's why we do contact tracing. So in contact tracing, what then will be the steps? We have identification of the contacts that we have above. We will now have, we categorize these contacts because they could be very close or further away from the patient. And then we notify them, we monitor them and collect specimens from them. In case some are infected, we manage the situation and then we implement preventive measures as well as document and follow up. And then we had our part 2, which led us to question 13, which is what are the biosafety and biosecurity measures? Of course, this based on the spread of the disease and everything, we will obviously need a biosafety level 3 and then level 2 for work as well. We'll need proper PPE, and then we'll need proper decontamination and disinfection procedures. For biosecurity, we obviously need to do access control, inventory management, and also conduct all experiments within biosafety level 3 and biosafety level 2 as well. What are the observations expected by the survey team? The survey team here was team from the Ministry of Health, team from the Ministry of Agriculture, team from the Ministry of Environment. So what then are they expected to see in the field? They are going to see the what. The what in this case is the disease. What is actually causing this outbreak? They are expected to get that. They are expected to see the who. Who are those infected? Who are those at risk? Who are those involved? They are expected to see the when. Okay, when did the outbreak occur and so on. They are also expected to get the where. Where particularly are we talking about? The location. Okay, and the why and the how. The possible causes, transmission routes, and contributing factors. So that is why they are expected to see human cases, high mosquito densities, dead or sick birds, environmental risk factors, okay, like stagnant water, poor waste management, poor human behaviors, and so on. And then what are the, question 15 says, what are the common methods for collecting of various environmental and mosquito samples? Common methods for collecting mosquito samples, we have the Center for Disease Control light traps, we have gravity traps, we have lava and people sampling, we have aspirator collections, pooling, and storage. For environmental samples, of course, we'll do environmental sampling like water sampling, collecting blood from animals, tissue sampling from suspected or even dead animals, as well as environmental vector to host interface observations. The next question says, list the laboratory investigations that were applied for diagnosis of the causative agent. So at this point, we already have the laboratory investigations that have been applied. So possible laboratory investigations that were applied, we have ELISA to check for the presence of IgM, IgG. We have molecular detection methods. We have PCR methods, that's the real time. We also have virus isolation and antigen detection investigations that were done. And the different precautions that were used, use of biosafety cabinets, use of proper PPE, and use of proper containment for virus culturing. What is the definition of zoonotic disease and arbovirus disease? Zoonotic disease is an infectious disease that is naturally transmitted between the vertebrate animal and the human host. And then arbovirus disease is a disease that is transmitted to vertebrate host by blood-feeding arthropods. Arthropods in this case, we mean mosquitoes, we mean ticks, we mean sandflies. So they are actually a subset of zoonotic disease that are involved with animal reservoirs. Example of these diseases, of course, we have West Nile, we have dengue, we have chikungunya, we have Zika virus. Question 18 says, what are the roles of mosquito, horse, and wild birds in the transmission of West Nile virus? So at this point, we have done laboratory investigations, and we have confirmed that our outbreak is being caused by the West Nile virus. Wild birds have the role of transmitting West Nile virus because they are able to sustain viral replication. They have high viremia, and then they are blood feeding. They have high viremia and infects mosquitoes that feeds on them. Okay, so that's why they are able to transmit the virus. Mosquitoes, on the other hand, will naturally go for blood. So in the course of going for blood, they will pick up the disease from the wild birds or from the animals or from the horse. And then horse is a dead end, meaning that the virus doesn't replicate within the horse. So they can develop severe diseases, but generally will not produce high viremia. So they are not responsible for transmitting the disease from one horse to the other or from one, or that is why at this point we can be able to establish that the disease transmission from human to human is very rare. Okay. And then, do you think the Ministry of Health managed this outbreak situation very well? We could say yes. Okay, thank you. We say yes, based on what? The rapid multi-sectoral response that was involved. Almost all sectors, within a short while, they're able to coordinate most of the sectors, the health, the environment, the agriculture, put together. Most times it's usually very difficult, but they were able to do that. So they tried. And then the prompt field investigation, as well as the comprehensive surveillance that was being done, data that was being collected. We have laboratory confirmation, as well as public health communication. So in all of this proves that they actually did a good job in managing the outbreak. We have our question 20 there, that what are the risk communication elements? Risk communication elements, you know, when there is an outbreak, there is a possibility that people can be misinformed, which can create fear, which can create unnecessary tension. People can even be, for example, in the times of COVID, when they say people should take COVID vaccine, some people within my area, some of them were like, no, that vaccine is a chemical that will help people not to reproduce. So they will not want to take the vaccine. So risk communication elements are those things that you are supposed to take into control or in place to make sure that the right information is being given to the right quarters. So you need to know the audience you're dealing with. And you also need to get feedback response from them. So in this case, our risk communication elements involve an integration of all parts. And it involves audience analysis and stakeholder mapping. It involves a clear, timely, and transparent messaging. Sometimes when there is an outbreak, people could choose to inflate values, create tension within the communities. They are inflating values because they want the government to keep pumping more money into their structure, so they can choose to inflate values. But a good communication, you have to be transparent with the values that you get. Or sometimes they don't want scandals, so they try to make it look like it's nothing. Meanwhile, it's actually something that is heavy. And then there's also this aspect of a two-way communication. A two-way communication means you are not the only one giving information to the population. You also have to put your ears on the ground to get what the population is saying concerning the situation on ground. So because it will help you to actually get feedback, manage, and then you re-inform them on what they could be having misconceptions about. Okay, so our question 21 says, in line with the international health regulations, international health regulations it's a kind of a frame that puts together regulations that binds WHO member states as to how to get back to WHO in case of outbreak. Okay, so it's actually a table where they put there if the disease is of high economic importance, if the outbreak is of high economic importance, if the outbreak is unusual and unexpected. Thirdly, if the outbreak can spread to neighboring countries. And fourthly, if the outbreak can affect international travels. So if there is an outbreak that ticks two of these that ticks two of the four, then you have to report to WHO within 24 hours of an outbreak. So with this, we notice that the outbreak we are dealing with actually has ticked like three of these. It's of major public health importance, affecting different sectors of the economy. It is unusual and unexpected. It can affect other international countries, considering that tourists are involved. So we have to inform WHO within 24 hours of the outbreak. And then our last question says, what are the recommendations to follow at all levels? When there is an outbreak, recommendations means you have somebody writing recommendations to a particular people. In this case, you notice that we have individuals that are involved. We have the public, we have the government. So we make recommendations to individuals or households. To individuals who tell them, please protect yourselves against mosquito bites, use repellents, sleep under insecticide-treated mosquito nets, and things like that. And then for the health authorities, we recommend that they should be very careful when dealing with samples. They should wear proper PPE and so on. And then to the vector control sector, they also have their own role to play, as well as every other. So the recommendations are given to different sectors based on the role they have to play. Even the veterinary sector, they have their own role to play in situations like this. So we have short-term recommendations and we have long-term recommendations. Short-term recommendations are to what? Enhance vector control, do personal protection and campaigns, we enhance animal surveillance, do healthcare preparedness, and do public communications to keep the people aware. And then for our long-term recommendations, we also do a One Health coordination that involves different sectors put together to address situations like this, especially in the future. So, thank you very much. Okay.
So the last group, NIB. Actually, we had another extra group, the NIB group, that they, just get the scenario today morning. They prepare half of them. They go on.
Oh, I said.
But they are not here. Anyone? Number? Can you cover?
And Miss Ready will play. Fine. Did you get it? Wait for a second. Let's see question. Okay. Do you want to? Okay.
The usual segment the final fight for that is sending. They have 10 or 15 minutes. I think it will be enough for you.
Yes, we have only prepared 10. Sky. वकार का ऑन था ना इसको तो मैं कुछ नहीं कर रही हूँ वकार का ऑन था ना नहीं नहीं हमारी भी आई नहीं है वकार का ईमेल आईडी ऑन था ना वो खोल मैंने भी भेजी नहीं है अभी इसको भी भेजी नहीं है And should I start?
Yes, sure.
Okay. Assalamu alaikum. My name is Faria Tariq and my group members include Aksa, Tuba, Wakar and Zehar. So the title of this case study is Emergence of a Febrile Illness of Unknown Causes Among the Population and Visitors of Upper Egypt. नहीं हो रहा
But you want that one arrow से आगे start होती
So let's move forward to the background. That's a brief overview. That it's a fictional case study for training purpose only, and it focuses on a febrile illness of unknown cause in Upper Egypt in the year 2002. Aswan was chosen for its climate, topography, and geography. So the data that's given in this study is based on data from applied research in Egypt, and the symptoms include fever, neurological and intestinal signs with high mortality. And the affected population are in contact with farm animals, especially equines and birds, or the people near the waterways. And the cases concentrated in Aspan and nearby governorates. This study highlights outbreak investigation steps, challenges, biosafety, and biosecurity concepts. Yeah. So if we talk about the epidemiological context, why Aspan was chosen? Because of its population, that's nearly 1 million people, and majority, that is the 56%, live in rural areas, and the livelihood depends on the animal and wildlife activities, and that is why there is high potential for zoonotic transmission. And if we talk about the tourism, there is close interaction between the tourists and equines through horse-drawn carts. So that's a possible human-animal contact route. And if we talk about the climate is hot and dry with rare rainfall. Occasional floods may spread contaminants through agricultural drains and river Nile. And if we talk about the environmental exposure, wastewater disposal into the Nile and seasonal floods increase risk of waterborne and vector-borne transmission. And because of the tourist influx, especially in the winter months, it may facilitate the disease introduction and spread. So the first question is, what are the factors that make Aswan Governorate an interesting area for epidemiological investigations? So these are the main factors. The intense human-animal interaction, the proximity to water sources, the cross-border traffic with Sudan and high tourism, and the climate factors like vector breeding and transmission. So we did not have time to prepare a questionnaire. So these are the few details that we included in it. We can add the identification that includes the name, age, gender, occupation, and address. The clinical details that includes the onset date, symptoms, and outcome. The exposure history that includes contact with animals, travel, mosquito bites, water exposure, and the sample info like type, date collected, test requested, and result. And few other things like treatment and follow-up. And you should link each sample ID to its corresponding case ID for accurate tracking and analysis. So if we talk about the clinical and post-mortem investigations that are applied to diagnose the cause of the cases. So in the clinical investigations, we have vitals, neurostat, blood tests includes complete blood count, LFT, RFT, electrolytes, and CSF analysis and culturing, and RT-PCR, viral RNA bacterial culture if needed. And if we talk about the post-mortem investigations, autopsy with organ sampling, histopathology, etc., could be used. So what are the recommended samples to collect from patients according to the mentioned symptoms? Blood samples could be collected, cerebrospinal fluid could be collected, whole blood sample could be collected, urine can also be collected, nasopharyngeal swab, tissue samples, ELISA, and you can also store the aliquots, and metadata can also be involved. If we describe and classify the different suspected causes that may lead to febrile illness symptoms of these cases, so it includes the viral causes, that includes the West Nile virus, Rift Valley fever virus, and bacterial possibilities could also be involved like Leptospira, Brucella, and parasitic cause could also be there, like Plasmodium species, and toxin-related causes can also be there, like environmental or foodborne toxins, but these are less likely. And what are the possible ways of transmission of this unknown disease? It could be mosquito-borne transmission, or human and horses can also become infected through mosquito bites, but these are dead end hosts, like it cannot be transmitted from human and horses. Blood transfusion, organ transplant, and laboratory exposure, as well as person-to-person or aerosol transmission can also be one of the possible ways. So what type of packaging should be used to transport these samples? We can use triple packaging. The first one is the primary container. It includes a leak-proof tube or a vial, and the secondary container to secure the the primary container, and the outer container that is rigid, protective package with biohazard label, address, and documentation. Which type of surveillance technique do you think must be activated and conducted during approach of the case, and why? So we can integrate surveillance system combining the human, animal, and vector data. So syndromic surveillance can be activated, and it includes to monitor febrile and neurological cases in hospitals and clinics, event-based surveillance, and active surveillance as well. So do you consider this outbreak as severe? How do you define this and why? So because over 100 humans were infected within a short period, and neurological complication and deaths were also involved. So this outbreak could be considered as severe, and because it can spread to other governorates and can infect other people, like foreign tourists as well. Thank you.
And let me move fast. Okay. So I'm going to announce the best answers as well. Maybe the item, where's the item at? I want to stop the recording. We're finished. Thank you.